Welcome, everyone, to the Genetic Engineering and Biotechnology News webinar series. Our presentation today is entitled Epitope Mapping in Antibody Therapeutics and Vaccine Development, the Evolving Landscape. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be your moderator for today's webinar presentation. Identifying the binding sites that antibodies recognize on their protein targets is critical to the discovery and development of new vaccines and drug therapies. Over the past several years, there have been a rapidly growing number of monoclonal antibodies tested for effectiveness in clinical trials. However, during the same time frame, the speed of epitope mapping techniques has often not kept pace with the increasingly large numbers of monoclonal antibodies being isolated. Additionally, with the large number of monoclonal antibody therapeutics being developed, and very often against identical targets, obtaining strong intellectual property protection for antibodies has become not only progressively challenging, but a paramount issue for the biopharmaceutical industry. Increasingly, the use of epitope mapping has become essential for demonstrating the patentability of therapeutic antibodies. Let's meet our panel so that they can provide us with their scientific and legal expertise into epitope mapping for drug and vaccine development. Our first speaker today is Joseph Rucker, VP of Research and Development at Integral Molecular. Dr. Rucker will introduce us to some of the principles of epitope mapping and describe the benefits of Integral Molecular's high-throughput epitope mapping approach. Dr. Rucker will be followed by James Crow, Professor of Pediatrics, Microbiology, and Immunology at Vanderbilt University Medical Center. Dr. Crow will discuss his data using epitope mapping to reveal antibody mechanisms of action, which is critical for the rational design and assessment of effective vaccines. And our final speaker today will be Lisa Hale, partner at DLA Piper and co-chair of the Global Life Sciences sector. Dr. Hale will update us on the current intellectual property landscape and what is protectable by patents with respect to antibodies and their epitopes. So before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We will try and answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. So without any further ado, let's get our presentations underway. Joe, take it away.
I want to take this chance to thank Jen for giving us the opportunity to talk today, and to the other speakers to providing their insights into epitope mapping. I'd first like to begin just briefly with introducing Integral Molecular. Integral Molecular, we're a biotech company located in Philadelphia, and really our core expertise is in antibody isolation and characterization, with a particular emphasis on epitope mapping. And over the last, many years, we've epitope mapped over 500 different antibodies for NIH, for customers, and for our own internal projects. I should note that many people know us actually for our expertise in complex membrane proteins, and our interest in epitope mapping really derives from that interest in complex proteins. So let's begin with why epitope mapping. So there are a lot of reasons why one would want to epitope map. My talk is going to be really focused on high throughput aspects of mapping. Why would you want to map epitope map lots of different antibodies? And that could be for differentiating candidate molecules as you're deriving therapeutic antibodies, or for selecting complementary combinations of antibodies for use in therapeutic cocktails. Of course, many people epitope map to really understand the mechanism of action of their antibody, and Jim will talk in great detail about that as it applies to pathogenic viruses. And then, of course, epitope mapping is a way that people understand their therapeutic product in terms of intellectual property and regulatory approvals, and that's, of course, what Lisa will talk about. But let's begin with what is an antibody epitope. When an antibody binds an antigen, they form a binding surface. The surface on the antigen is what is referred to the epitope, and the residues of the epitope are what are interacting with the antibody. Now, not all of the residues are energetically contributing to the binding interaction, but only some of them are. These are referred to as the energetic hotspot residues. Sometimes I'll refer to them as the critical residues. And so in understanding an epitope, you not only want to understand the residues that are on the surface, but the residues that are contributing to the binding. Another aspect of epitopes, which is important to point out, is the difference between linear epitopes and conformational epitopes, also sometimes referred to as discontinuous epitopes. In the structure in green is an example of a linear epitope. A linear epitope is where the residues contributing to the epitope are continuous in the sequence. On the other hand, for many epitopes, particularly those involved with therapeutic antibodies, the epitope is formed by residues that are discontinuous on the sequence, but on the structure come together in 3 dimensions to form the epitope. And this is what's referred to, as I said, as a conformational epitope or as a discontinuous epitope. And the distinction between linear and conformational epitopes has ramifications for how you epitope map, and as Lisa will say, also has some ramifications for legally for intellectual property. So there are a number of different epitope mapping technologies out there, and they all have they're all advantages and disadvantages to all of them. What are the types of information that epitope mapping technologies can give you? Well, some of them can answer questions about conformational epitopes. Some of them are only good for linear epitopes. There's questions about resolution. Some of the techniques are good for getting atomic resolution or residue level resolution, and some are actually somewhat lower resolution to give you areas of interaction. Some can do complex proteins. Some are more limited to simpler proteins. Does the technique give you energetic information, or is it just giving you the overall structural information? And finally, of course, which is important for everybody, is how long does the technique take? So our technology, which is shotgun mutagenesis epitope mapping, which is a comprehensive mutagenesis technique, we're interested in trying to hit all of these different aspects of epitope mapping, trying to get high-resolution epitopes on complex proteins, getting energetic information, but getting it in a rapid fashion. And I'll talk to you a little bit about how we do this and then what you get from that type of information. So let me give you an overview of this technology. I'm not going to go into much detail, but if you have any questions, feel free to answer in the Q&A session or to contact us offline. But what we do is when we start comprehensive mapping, the high-throughput mapping, we engineer a parental plasmid that's optimized for mutagenesis and also optimized for high expression. We can then create a comprehensive alanine scan or really any type of mutational library of hundreds to thousands mutations in under about a month, so it's a very rapid process. Obviously, if we're doing these types of analyses, we need to use robotics and high-throughput methodology such as flow cytometry to do the analysis, and generally in 3D 4-well plates. So we can collect a lot of data very, very quickly. Of course, when you have this high density of data, you need to have software and algorithms for both tracking the data and for analyzing the data. And the goal is to integrate these epitopes that we can pull out from this platform and to do structural analysis very rapidly in real time. So the idea is that we're doing comprehensive mutagenesis and expression in human cells, and we're mapping conformational or linear epitopes on really any type of protein. It could be simple, but it could be complex membrane proteins, viral proteins, membrane proteins, etc. At this point, I want to transition over to talking about specific examples that we've used our high-throughput mutagenesis technology on, case studies, to give you a sense of what we can do and really what type of information this epitope mapping can bring to you. So this first case study is going to be a comparison of our epitope mapping technology, shotgun mutagenesis, with co-crystallography. And this is of a viral envelope protein, in this case, the Ebola GP protein. In the left structure, you can see the structure of the Ebola GP protein, and in blue are the epitope that is derived from co-crystallography. These are residues that are potentially contributing to the interaction energetically, but are just found in the footprint of the antibody. Going to the center panel, in green, these are the residues that we identified as the energetically critical residues. They lie within the crystallographic footprint, but in fact, these are the residues that are key for determining the affinity of the interaction. Now, of course, with any type of study, you want to do some confirmatory functional studies. In the case of viruses, we can do neutralization escape. And here you can see an example where we've taken the residues, the residues in orange, which are within those energetically critical residues, allow for neutralization escape. Obviously, for other types of proteins, you can do other types of functional conformations as well. Another use of epitope mapping, of high-throughput epitope mapping, looking at multiple antibodies, is figuring out ways of combining antibodies such that they make better therapeutics. And here's another example using Ebola. So there are 7 antibodies that we mapped. And in the left panel, you can see that these antibodies map to a couple of different regions on the Ebola glycoprotein. Now, there have been several cocktails of Ebola antibodies that have been tested both in animals and in humans during the recent outbreak. And the first cocktail, shown in the center, all of the antibodies within the cocktail were binding to the same region of the Ebola protein. And in this case, this cocktail provided no survival advantage. On the other hand, on the right is the cocktail that was actually used during the recent outbreak, and we mapped those antibodies that are part of that cocktail to multiple regions of the protein, and suggesting that you need to hit multiple regions of the protein in order to get efficacy. So figuring out the epitope of each of the component of a cocktail really provides insight into how to optimize those cocktails for therapeutics. In another case study, we looked at another virus. In this case, it's chikungunya virus, and we mapped many antibodies. In this case, I'm showing 15 antibodies, some of them neutralizing, shown in gold, some of them non-neutralizing, shown in green. And we can get a sense from this about those regions of the protein that are important for neutralization. We, of course, can use this to identify complementary mAbs that could be useful for cocktail therapeutics. But we could also get more detailed information on mechanism of action. And on the right, this is an antibody which we were able to show was potentially binding across the monomers of this complex oligomeric protein, really suggesting an unusual, an interesting mechanism of action. And this type of information was put into a paper and also into a patent. So this type of high-resolution epitope mapping helps elucidate antibody mechanism of action. So it looks like we only do virus proteins, but you can do many types of proteins with this high-throughput mutagenesis technology. And this is a list of all of the case studies that we can talk about. Some of them are, of course, viruses. Some of them are, on the other hand, cellular membrane proteins. We've done GPCRs, 4TM proteins, such as the claudins, or a number of different other membrane proteins, which have interest from for example, for oncology, such as HER2. And really, it's a wide range of different protein sizes, everywhere from library sizes, everywhere from a couple of hundred residues to over 1,000 residues. So we can really not limit it in terms of the type of proteins we look at or the size of the protein. So let's go into a few case studies here. So in terms of differentiating therapeutic antibodies, here's an example, GPCR, CXCR4, and this was work that was done by Ablinks and was later published in PNAS and used for patent filings as well as regulatory filings. And what we showed is that the 2 nanobodies, shown in blue and red, saw an epitope that was distinct from epitopes that were found for other CXCR4 antibodies, and in fact were distinct from each other as well. And this information was what was used to help decide to move these antibodies forward into the regulatory process. In another example, this is an example of HER2, which of course is an important oncology target. We had a customer who was interested in mapping their antibody in comparison to a therapeutic antibody. We mapped the therapeutic antibody shown on the right. You can see the three red residues are defining the critical residues for this known therapeutic antibody. And we were able to differentiate this customer's antibody from the therapeutic antibody. And this helps to differentiate their antibody both in terms of intellectual property and also in terms of mechanism of action. So that was a very nice case study for us. And then finally, this is back to another GPCR, CXCR2. This is a project we did for Novartis. They used this both in a publication and also within a patent. We mapped 2 nanobodies for this customer, for this partner. And what you can see is one of the antibodies in beige is a linear epitope on the N-terminus of the protein, while the other antibody is actually a rather complex conformational epitope shown in red, utilizing 2 loops which obviously are coming together in the overall structure. And this information was being used to differentiate these 2 antibodies so that they could know that they would be complementary when forming a bispecific antibody, and it's ended up being a very interesting case study for why epitope mapping. We've been able to high-throughput epitope map actually at a very rapid rate, upwards of 20 epitopes per month. And this has been done through optimization of the mutagenesis protocols, of the analysis, and of the actual analytical procedures. And this approach then enables us to really map large collections of antibodies, which is often important if you're trying to understand, for example, the neutralizing antibody response against infection. So I want to just finish with an example where we took a large panel of antibodies, in this case against the dengue virus, over 100 different antibodies. We were able to map all these antibodies rapidly, look at their epitopes, compare their epitopes, and use this information in a variety of different papers shown on the right, looking at many aspects of antibody neutralization, of virus mechanism of action. And in fact, some of these papers go beyond epitope mapping to doing everything from functional mapping to protein engineering. And these papers can be used to really give you a good example not only of what our shotgun mutagenesis technology can do, but can give you an idea of how epitope mapping can be useful for you. And at this point, I thank you for your attention and look forward to answering your questions. Thank you.
Thanks, Joe. That was a great presentation to get us started, and you provided us with some really nice insight into the complexity surrounding epitope mapping, as well as showing everyone the benefits of Molecular's high-throughput approach. So we thank you for that. Before we move on to Jim's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentation. Simply type your question into the Q&A box on the left-hand side of the screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. So let's get our next presentation under the way. Jim, the floor, as they say, is now yours.
Thank you, and it's great to join you all today. I'm going to be speaking today about mapping the epitopes for human monoclonal antibodies directed to viruses. And we're interested in this for several reasons. One, human monoclonal antibodies can be used as therapeutics themselves against viruses or used as prophylactic preventative treatments, but they also can elucidate the targets for neutralizing antibodies that could be incorporated in vaccines. So when we understand the interface between an antibody, as seen here on the left in colors, and a viral antigen, in this case hemagglutinin, surface protein, when we understand the interface and the intricacies of that, we can use that information to develop either the antibody or the antigen as a vaccine. So that's the rationale for what we're doing here. I'm going to give you some general principles about epitopes on viral antigens. First, these epitopes are not simple peptides, or they're generally not linear features. They're typically very complex surfaces. So I'll show you some examples of epitopes that are comprised of multiple loops in a protein within a single protomer of a protein. And protomer is the structural unit of an oligomeric protein. It's really the smallest unit, composed of at least two different protein chains that form a larger heteroligomer by association of two or more copies. So some antibodies recognize a single protomer, but they usually recognize multiple loops in that protomer. But then if you have an oligomeric protein, you can see multiple protomers in that oligomer. And in a particle, like a virus particle, often you'll have thousands of copies of an oligomer on the surface, and antibodies can recognize multiple oligomers. And once we understand a trimeric protein or dimeric protein, whatever the oligomeric form is, and we've studied it as a soluble factor, often we will find antibodies recognize things differently than when they're in a viral particle, when you have multiple copies or configurations. And in that setting, you can even have issues of accessibility that regulate recognition of antibodies. So let's look at some examples. First example is from influenza. So on the left, this is a space-filling structure of the Influenza H2 from the 1918 strain that killed about 50 million people on the Earth. And we obtained human monoclonal antibodies from individuals who were originally exposed to this virus when they were about 2 years old, and they are currently about 100 years old. And by isolating B cells from those individuals, we found 1918 specific monoclonal antibodies. So this is an example of, on the top, you see 3 copies in yellow and red of a human FAB in complex with the space-filling HA. And this structure was determined by crystallography by Ian Wilson's group at Scripps with our antibody and their proteins. You can see on the top right the surface projection of the footprint of the antibody. You can see it's quite large. There's an outer zone in yellow, and then a central zone that's in a purple color. And this is quite a large footprint, and it's made up of multiple loops in the HA protein. And this work was quite interesting because we isolated these antibodies in 2008, and the next year another H1 flu pandemic occurred, the 2009 H1 pandemic that you're familiar with from the news. And by looking at the epitope, we predicted that these antibodies from 1918 survivors would cross-react and, in fact, kill the 2009 virus, which is what we're showing on the bottom right. And this predicted that elderly people would be relatively immune from the 2009 pandemic because of their prior immunity in 1918, and that turned out to be true. So even from an epidemiology standpoint, using epitope information can sometimes be interesting. So that's the first major pandemic of 20th century. In the next slide, we'll see in 1957 pandemic, which was an H2 pandemic virus, and we've done the same thing. We've isolated human monoclonal antibodies from survivors of the H2 1957 virus, and these are now 3 different crystallography-derived structures at atomic resolution, and you see in the sort of blue and gray colors at the top 3 FABs again. These antibodies all bind the top of the hemagglutinin molecule. On the bottom, underneath those structures, you see in yellow the footprint of those antibodies. And when you compare each of these 3, you can see they have a similar general footprint, but if you inspect the top structures, each of those antibodies is coming in at a different angle. So what we learned in those studies is there's not a single approach or pose or angle of the antibodies, but the antigenic site or the epitope is very similar, and there are critical residues in the center of that epitope that need to be engaged. And in fact, this footprint that you're looking at is essentially the receptor binding site. So these antibodies, we know their mechanism is to block attachment because they bind to the receptor binding site and they mimic the sialic acid receptor. So that's a case where we not only understood the epidemiology from these structures, but also the mechanism of neutralization based on the structure. Next case is a different virus. This is Marburg virus, which is a filovirus similar to Ebola virus. So we studied a traveler who went to Uganda and went into a cave filled with fruit bats, and a few days later, the subject became very ill, ultimately survived, and it was discovered that this person had acquired Marburg infection in Uganda. So we isolated over 50 human monoclonal antibodies from this survivor and tried to determine where these neutralizing antibodies bind on the Marburg glycoprotein. At the time, it's not really possible to do crystallography complexes because even the glycoprotein structure had not been crystallized. So this data I'm showing you, these data here are from single particle analysis. And single particle analysis is a group of related sort of computerized image processing techniques that are used to analyze images from transmission electron microscopy. So you take lots of TEM pictures, which are not very high resolution. They're just fuzzy little pictures. And using these computational methods, these were developed to improve and extend the information that you can get from TEM images of particulate samples like this, proteins on viruses. The individual images of stained particles are very noisy, but then you can combine digitized images of hundreds or thousands of these similar particles together, and they give an image with a stronger and more easily interpretable structure or features. And in that case, you can build up a three-dimensional reconstruction of the glycoprotein, which is a trimer, and in this case, you can, again, see 3 FAB copies on the Marburg glycoprotein. This was really the first structural information we had about the Marburg glycoprotein, and we could see where the neutralizing antibodies were binding. So these are 3 similar antibodies. They all end up at the same place, but just like the H2 antibodies for flu I showed you, they each come in a slightly different angle or twist or binding pose. That's quite interesting. We had another set of antibodies that came in a flatter angle, so these are in 4 different colors, each 4 different additional monoclonal antibodies. But again, they're all binding to the top of the gray Marburg glycoprotein here. And so if we overlay all of that information, it allowed us to understand where the neutralizing antibodies are binding. And this is just a cartoon representation of the glycoprotein. So there's a viral membrane on the bottom, and then the structure, and the arrows with colors are showing you either steep or shallow binding of neutralizing antibodies that we obtained. And then there's a top view on the right showing there's various angles at which these antibodies approach, but ultimately they all bind to the same major antigenic site. There's minor epitope differences between them, but ultimately they're binding the same site. And using this information, we determined that these antibodies are binding, again, the receptor binding site, which for Marburg is called Niemann-Pick C1 protein. So this was the putative binding site, and subsequently this has been proven with crystallography by Erica Safire's group. So this allowed us to determine the epitope recognition using this single particle EM reconstruction. And all these reconstructions that I'm showing you were done by Andrew Ward's group at Scripps with antibodies that we isolated from this survivor. The next technique is also a low-resolution technique, but very helpful. And this is called small-angle X-ray scattering technique. And so in this case, I'm showing you the Marburg glycoprotein on the left and the Ebola glycoprotein on the right. And what was interesting when we obtained these Marburg human monoclonal antibodies, some of them bound to a cleaved or a processed form of the Ebola glycoprotein, but they wouldn't bind to the virus and they wouldn't neutralize the virus. So they were cross-reactive, but they only neutralized Marburg. And I've already shown you the Marburg antibodies bind at the top of the glycoprotein. One of the problems with the EM structures that we had in the crystallography that we did with Erica Safire was that there's a domain called the mucin-like domain. And in these images that you're looking at, the gray envelope is the small angle X-ray scattering data, and the wings hanging off are structures that you can't see in the crystallography or even in the EM, really. The wings are the mucin-like domain, and what the small angle X-ray scattering revealed was that in Marburg glycoprotein on the left, these domains are hanging down, they're displaced to the side, and they are not blocking the receptor binding domain at the top, and therefore these antibodies could bind. Whereas on the right, in Ebola glycoprotein, this mucin-like domain is arrayed at the top, pointing upwards, sort of like an antenna, but it sits right over the receptor binding domain, and that explained to us why the Marburg antibodies could cross-react with cleaved forms of glycoprotein, but they could not neutralize the virus. So again, giving us mechanistic information about how these antibodies were working and cross-reacting. All right, I'm going to now shift to a different technique, and again, another low-resolution technique, and this is called HD exchange. It's hydrogen deuterium exchange. Some people call this HD exchange. Sometimes they call it DXMS for deuterium exchange mass spectrometry. A lot of nomenclature, but I think most people would call it HD exchange. It's a chemical reaction in which you, in our case, we're putting proteins in solution, and you get a covalently bonded hydrogen atom that is replaced by a deuterium atom, or vice versa. So if you put proteins in the presence of deuterium, you'll get exchange of the hydrogens. Usually the examined protons are the amides in the backbone of the protein, and so the method gives information about the solvent accessibility of various parts of the molecule and thus the tertiary structure of the protein. So the residues that label with deuterium or exchange are on the surface. And so this is a case of a virus called rotavirus. It's the most common cause of diarrhea in children, or severe dehydrating diarrhea. And this protein is called the virus protein 6. So there's 3 protomers that, again, make a trimer in the virus, and you're looking at a space-filling model of the trimer. And the data from the HG exchange, the mass spectrometry, is on the top. So you see a continuous protein sequence, but it's clipped into little segments, and that's where the digestion for the mass spec cut the protein into peptides. And if the peptides labeled well, they are shown in red, and if they labeled poorly, they're shown in blue. And there's different times of 10, 100, 1,000 seconds. So that's what the 3 lines are for each case. And we labeled the VP6 protein alone, and then the VP6 protein with the FAB. And by doing that, we can subtract the signal and see what disappears. And what disappears is shown on the space-filling model in orange. And what we discovered was that the labeling that disappears in the presence of the FAB is the footprint of that antibody. And remarkably, the antibody crosses 2 protomers. So region A is in one protomer, and region B is in a separate protomer. So the epitope, the antibody doesn't really care what protomer these features are coming from. It just sees a surface, a continuous surface. But in fact, they're coming from 2 different protomers. So that's an example, again, of 2 protomers comprising an epitope. So this is HD exchange. Another technique would be cryo-EM. So cryo-EM is freezing particles in ice and taking images, often with different tilts, and by doing that, you can get a 3-dimensional reconstruction. So this is the same antibody that I just showed you in the HD exchange, but looking at it with cryo-EM, and we're looking at a particle. Those particles have either 5 copies at the top left of VP6 trimers, and there's different channels on the particle. In some cases, in the middle of the right, type 2 or type 3 channels on this particle have 6 oligomers that are trimers of VP6. So it's very complicated when you look at the particle. And in each case, the epitope is the same on these proteins, but depending on where that oligomer is placed in the context of a 5 or 6 copy unit, on the particle, you'll get a little bit different binding. So we could only discover this by doing cryo-EM because we needed the particle. So there's the case where you see it in deuterium exchange or HD exchange to get the footprint, and then we fit the crystal structures of the individual elements into cryo-EM and got essentially a atomic resolution structure. Here's another example of cryo-EM. This is work we did with Shimei Locke in Singapore, and her group did the cryo-EM. And you can see the full particle in the top right in a greenish color, and then the human FABs to dengue virus are attached in blue, the spiky figures. And dengue is quite interesting because the surface protein is a dimer, and yet those dimers array into what are called rafts. So what you're looking at in the main part of the slide are blue, yellow, red dimers. 3 of those dimers make a raft. So you're looking at a raft. And when we did the cryo of this potent neutralizing antibody for dengue, what's shown in green on the left is the footprint of that antibody in the cryo. And on the right is just another view. It's a ribbon diagram, and the gray space filling sitting on top is the FAB seen in the cryo. And in this case, if you look closely, this antibody is seeing 5 different domains. There's 3 different yellow ones and a blue one and a red one. Those are the domains. And those are present in 3 different protomers of the surface protein of dengue. And in fact, it's crossing 2 dimers. So this is perhaps the most quaternary or complex epitope that we've stumbled into. And again, the antibody doesn't really care whether these proteins are coming from a protomer, a dimer, or a raft. It just sees the surface. So here, just outlining the 2 features that are the protomers within a dimer in those orange boxes. So that's 1 dimer but 2 protomers. And so the cryo, we can pull apart and see the protein. So another benefit of cryo is you can get conformational changes when proteins go into particles, and we can see that. So this is another dengue human monoclonal antibody footprint. What you're looking at is 1 protomer of the dengue surface protein with the 3 domains, again, yellow, red, and blue. But the footprint of the antibody is in the cyan color. And in the crystal structure of the soluble protein, this protomer is linear, like the straight horizontal line. But in the cryo, where we pulled this out, it's actually at a pretty acute angle. And so the footprint is in the angle. And in fact, this antibody only recognizes the surface protein in the context of the particle. If you make this protein soluble, it becomes linear, and the human antibody does not even recognize it. So we would not even have been able to find this antibody if we had not used particles. So we've done a lot of work on dengue. We've isolated about 400 antibodies, and we can't do the crystallography or EM studies on that many antibodies. So this is an example of a case where we got 120 dengue monoclonal antibodies from a single individual, and we needed a high-throughput technique for doing this. And this is work we did in collaboration with Integral. You just heard Joe talk through that methodology. They made a library with an alanine scan or an alanine mutation at every position that would fold in the dengue surface protein. And you can again see the red, yellow, blue domains, and these are in dimers. So each dimer is the footprint of showing the footprint of a single monoclonal antibody. And the little green balls are space-filling residues that drop out binding. So we were able to identify the major antigenic regions, the fusion loop, domain 1, 2, 3, the PRM region from a single individual. This isn't possible to do this many antibodies using the other techniques I've talked about. And finally, you can combine techniques. So this would be called a hybrid method. This is a paper we published a year and a half ago or so, in which we did the single particle EM, and you can see the envelope of that on the left, where H5 hemagglutinin is sort of the bottom left of the image, and the top right, the donut-shaped thing, is a human FAB that's interacting with the H5 hemagglutinin. So that's the EM. And then we also did the hydrogen deuterium exchange, or DXMS, And on the right, you can see that peptide we identified in blue. We also knew the antibody blocked receptor binding, which is shown in orange. And then we got the crystal structure of the FAB, which is the ribbon diagram you can see in the donut shape on the left. And we put all this together computationally using a program called ROSETTA. So we computationally docked within the constraints of all these experimental data. And you can see that the footprint of the FAB sits right along the DXMS peptide, or the HG exchange identified peptide, and the receptor binding domain, which made sense to us from all the biological data we had. Interestingly, this antibody not only kills regular H5 flu, but also H5 flus that had been altered in the lab to be highly transmissible, that would go not only from bird to people, but also potentially people to people. There was a lot of concern about these viruses, and the data that I'm showing you here shows that people who are vaccinated with conventional H5 vaccines make antibodies that would also kill these viruses. And the reason they do that is they don't really touch the green residues on here, and the green ones are the highly transmissible residues. So this was important data. These are important data because it predicted we have vaccines for these unusual viruses that have been generated. So that's just a case where we understood policy and the potential efficacy of a vaccine really before a challenge was done, because we saw the antibody footprint differed from that of the variant. So that's a survey of various techniques that we've used in a broad categories using imaging or biochemistry and so on. And what we see is that viral targets are very complex. You really can't use one technique to get everything, and you need a diverse set of tools to do that. So thanks for spending time with us.
That was great. Thanks very much, Jim. I think our audience now has a great sense of how critical epitope mapping is towards the rational design of neutralizing antibodies. So thanks very much. So it looks like we're already starting to get some really great questions coming in. So I'm encouraging the audience to keep them coming. So let's begin our final presentation of the webinar. Lisa, when you're ready, you have our attention.
Thank you. I'd like to speak to you today about some patent and regulatory strategies around antibodies and epitopes. And you've already heard quite a bit about epitope mapping and identification of epitopes, which provides a basis for much of my discussion here today. As an overview, what I am going to discuss is, first, I'd like to get everyone on the same page and talk about what patents are and the standards for patentability. Then we'll talk about freedom to operate issues and the importance of epitope data for patentability and freedom to operate. We'll talk a bit about written description and the requirement for patents and how epitope identification falls into identifying written description. And then we'll talk about some recent life sciences court decisions that affect antibody and epitope patents. I'll provide some examples of issued patent claims around epitopes and antibodies, and then we'll talk at the end about the importance of epitope data for regulatory and biosimilar strategies. So first, to get everyone on the same page, we just want to briefly review what a patent is. The authority for patents, it comes from the Constitution, and the patent grants owners the right to exclude others from making, using, offering for sale, selling, or importing the claimed invention. The important thing to remember about a patent is that it provides a negative right to exclude others from doing what is claimed in the patent. The patent is not a right to do what is claimed in the patent, and we'll talk further about that in a few minutes. There are essentially three criteria for patentability in the United States, and countries outside the U.S. have very similar standards. First, you must show that your invention is useful and has some utility. Second, the invention must be new or novel. In other words, it couldn't have been known or used by others, either in the U.S. or abroad, and could not be in public use or sale in the U.S. more than 1 year before filing a patent application. The U.S. still has this 1-year grace period where a disclosure does not preclude you from obtaining rights in the U.S. However, if you're interested in foreign protection, you cannot have disclosed your invention prior to filing the patent application. The third criteria is called non-obvious or non-obviousness. This is where the differences between the invention and what was already known in the prior art would not have made the invention obvious to one of skill in the art at the time of the invention. A patent in the U.S. has a term 20 years from the date of the earliest effective filing date. So if you file a series of continuation or divisional or continuation in part applications, you still look to the earliest filing date to calculate your 20-year term. Next, we'll talk about freedom to operate and patents. A patent, again, gives the owner the negative right to exclude others from making, using, selling the patented product or method. So again, owning the patent does not ensure that you have freedom to operate. An example in the antibody or epitope space would be if company B obtained a patent for an antibody that binds to a very specific epitope on the EGF receptor. It's possible that they still could be blocked from using their antibody and using their invention because there may be a dominating or broader patent obtained earlier by company A, where company A obtains a patent that claims isolated antibodies that bind to EGFR. In this way, having the patent does not give you the right to do anything with the invention, and you may actually be blocked by a broader patent. So what is the importance of epitope data? In examining patent applications in the United States, there's a further criteria that is called written description and enablement, and this falls under USC Section 112. In order to obtain a patent, you have to show that you were in possession of the invention that you are claiming. And so in the antibody space, having binding data or epitope data will help you meet this 112 requirement to obtain your patent. The knowledge of your epitope distinguishes an antibody from prior art antibodies, and so you may be able to obtain a patent to an antibody broadly if the binding sites are not the same, if the epitope binding sites are not the same. You want to include information on conformational epitopes where possible so you can distinguish from known linear epitopes or epitopes that may have variant or mutant sequences in a particular disease indication that might distinguish from a wild type or a known prior art antibody. The knowledge of epitope binding can provide freedom to operate support to distinguish between novel antibodies and prior patented antibodies. And again, you might be able to obtain broad claims to your antibody if you can show that it's novel by its binding site or the epitope. As far as written description in patent law, the well-known case that defines written description is the Celebrex case. And this is the case where University of Rochester filed and obtained patents on methods for inhibiting COX-2, but not COX-1, using nonsteroidal compounds. The problem with the Rochester patent was that the patent application, when filed, did not include any chemical structures for examples of nonsteroidal compounds that could inhibit COX-2 but not COX-1. And so the courts found that the patent was invalid. This is a good example for looking at antibody and epitope applications. It tells you that if you can clearly define the epitope to which an antibody is binding and put that information in your patent application, it will increase the likelihood that you will meet the 112 requirement during examination of the application and also be able to defend your patent should you be sued in court. There have been several very important court cases, many of which were through the Supreme Court, that have affected life science inventions, and in particular will affect antibody and epitope patents going forward. And I'd like to talk about a couple of these in the next few slides. The first one, which was in 2012, is a Supreme Court decision generally referred to as Prometheus. And this is a case where the claims were directed to the administration of a drug to a patient and then observation of the levels of the metabolites of the drug in order to determine the next dosages of the drug. The courts found that these observations of what happens to a drug when you administer it to a patient is merely a law of nature. And this court found that just making these routine observations and going through routine steps does not take you to the level of a patentable invention. This case has been very important for looking at, for example, screening assays or diagnostic assays. In particular, if you've got a method of measuring the level of a biomarker or protein, for example, using antibodies or using epitope information, those methods will need to have novel steps or novel reagents used in these methods. So, for example, even using a novel antibody with a straightforward ELISA test will likely not be patent-eligible subject matter going forward. The next case that many of you have heard about, referred to as the MYRIAD case, also affects patents to antibodies and epitopes. This case briefly found that DNA is naturally occurring and a product of nature, and consistent with the Prometheus case, said that this is no longer patent-eligible subject matter. The court did hold that if you manipulate the DNA or the product of nature, for example, obtaining cDNA where you cut out the introns, you could still be eligible for patent protection. So how does this affect antibody and epitope patents? What we found now is that this case has been further reaching than we thought, and not just for genes per se. The U.S. Patent Office has been applying the case far beyond DNA sequences to include naturally occurring proteins. However, after the MYRIAD case, we have seen fragments of DNA or fragments of proteins, such as epitopes, still being patentable and issued under certain circumstances where the epitope, for example, was labeled with a detectable label or bound to a solid support, in which cases those would not be naturally occurring per se. The next case is actually a software case, but for the first time in December 2014 was applied to a life sciences related case and will have an effect on antibody and epitope patents going forward. What this case did was essentially confirm a test that came out of the Prometheus case that has two very important questions that are being asked both in court cases and by the USPTO in examining antibody and epitope type of patent applications. The first question, are the claims at issue directed to one of the patent ineligible concepts, for example, laws of nature, natural phenomenon, or abstract ideas? In our case, directed to proteins, antibodies, epitopes, or methods of identifying levels of proteins in a patient. The second question, if yes, is there an inventive concept? Do the claims include an element or combination of elements so the claims amount to significantly more than a claim to the ineligible concept? And this phrase, significantly more, is what the courts in the U.S. Patent Office are focused on. So let's look at the case where Alice was applied. And this is a case where MYRIAD, shortly after the Supreme Court case for MYRIAD, sued several companies because they were putting diagnostic kits on the market for detection of BRCA1 and BRCA2. So MYRIAD was alleging that there was infringement of several of their patents, and the Alice case was applied in this case to show that the claims set forth well-understood, routine, conventional steps that would have been engaged by scientists or one of skill in the art at the time the MYRIAD applications were filed. So, for example, an ELISA assay may have been run to detect levels of protein. In this particular case, it was, of course, looking at wild-type BRCA1 versus a patient's sample, but you can make the analogy to proteins and ELISA assays. The scope of patent-eligible subject matter following this case may be significantly narrowed. The composition claims to primers and probes in the MYRIAD case were found to be invalid, which leads one to an inference that going forward, protein fragments, including epitopes, may be patent ineligible subject matter going forward. Because this case was just decided in December, we are only now starting to see rejections from the patent office along these lines. But I believe that trying to claim a very simple linear epitope will likely cause you to get rejections from the U.S. Patent Office. And it's unclear where prior issued patents in this subject area will lie, whether they will be found to be invalid in the future. I just wanted to provide a few examples of issued claims in this area, and I'll run through them quickly just to make the point. But all of these examples show that either antibodies or epitopes are being claimed in a way that may likely provide, quote, significantly more in what the patent office is looking for in a claim. So, for example, in this first slide, we've got antibodies that recognize a conformation-dependent epitope, and there are sequences for the CDRs of the antibody. This may be enough information to take us into the patent-eligible subject matter. And in Claim 2, I highlighted chimeric antibodies and humanized antibodies, which are not normally found in nature and therefore may be patent eligible. In the second example, much of the same, where a monoclonal antibody is being claimed with respect to very specific epitope data. And again, in Claim 6, you'll see that the antibody is coupled to a therapeutic or diagnostic agent, which is something that would be non-naturally occurring. The next example, very similar again, I think I'll point to Claim 11 here, where the antibody is covalently linked to an additional functional moiety, again, taking it out of that non-naturally occurring subject matter. And then the final example is actually to a conformational epitope, which is described in a way where it's affixed to a support surface. Claim 6 is chemically bound. Again, these are ways to describe what might be considered a naturally occurring sequence of amino acids in a way that takes it into the patent-eligible subject matter. I'd like to move in my final section to regulatory considerations regarding epitopes. The FDA has provided guidance, which is an overview of FDA's approach at looking at antibodies when an application is submitted. And epitope data, while not required, clearly will help support some of the other studies, such as immunoreactivity. And as I've highlighted here, binding assays of the antibody to purified antigens and defined regions of antigens should be performed as feasible to determine affinity, avidity, and immunoreactivity. So again, while identification of the epitope or defining that epitope is not an absolute requirement, it will help support an application for an antibody for approval at the FDA. To use epitope data, this will be helpful to define the antibody binding specificity and, of course, distinguish it from other antibodies, provide a basis for limited cross-reactivity with other antigens, for example, showing an absence in non-target tissue. And the knowledge of epitope distribution can provide greater understanding of potential in vivo toxicity. And finally, I'd like to talk briefly about the value of epitope data for biosimilars. Again, I talked a little bit about the FDA guidance, and what the guidance provides is FDA's approach to determining biosimilarity, and it discusses important scientific considerations and data that will be necessary to demonstrate biosimilarity. An application that's submitted to the FDA must include information demonstrating that the biological product is biosimilar to the reference product based on data that may be derived, for example, from analytical studies that demonstrate that the biological product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components. The FDA has recommended a stepwise approach to demonstrating biosimilarity, and it can include a comparison of the proposed product to the reference product with respect to looking at PK, PD, immunogenicity, et cetera. And the FDA is looking at the totality of the evidence that's provided by the sponsor when looking at biosimilarity and evaluating biosimilarity. The applicant for the biosimilar application will use meaningful fingerprint-like analyses, which will include an understanding of the epitopes. Further analyses can be done through functional assays, such as binding assays, and again, epitope information is critically important toward this end. The final requirements for biosimilar applications are still being developed, and the FDA continually puts out updated guidance. But several of these have been published, as I mentioned. The FDA does expect that a sponsor will extensively characterize the proposed product and the reference product using state-of-the-art technology, because extensive characterization of both products serves as the foundation for a demonstration of biosimilarity, as I've mentioned earlier. The results of comparative analytical characterization will lead to one of four assessments within a development phase continuum. Either the reference product is not similar, similar, highly similar, or highly similar with fingerprint-like similarity, as I mentioned in the prior slide. Again, epitope data is not currently a requirement, but will clearly aid in defining the highest level of similarity, which is the goal. I'd like to just summarize in saying we talked about the value of epitope data for meeting the requirements for patentability, including written description and enablement under Section 112. We talked about the fact that the knowledge of epitopes can distinguish a novel antibody from prior art antibodies. The knowledge of epitope binding provides freedom to operate support to distinguish between a novel antibody and prior patented antibodies. Epitope data may be patentable, which will lead to new and valuable IP for a company. And with respect to biosimilars, while not absolutely required, epitope data is valuable for fingerprint-like analyses, binding studies, and comparison between products to identify the level of similarity. Thank you very much.
Excellent. Thank you, Lisa. You provided our audience with important updated information that many scientists often don't get to hear, but is a vital part of the drug development process. So before we start the Q&A, I would like to let everyone know that this is your final chance to submit questions, so hurry up and send them in now. All right. Since we have a bunch of good questions already rolling in, let's begin the Q&A. Keep the questions coming, though, and we'll try to get to as many of them as we can.
Okay, everyone, so looks like we're going to start the questions. We got a bunch of questions rolled in, and the first one's going to be for Joe. Joe, one of our audience members, would like to know, how do you ensure functional and structural integrity of the many mucins that are maintained?
Great, that's a great question. So, when we're doing the epitope mapping and we're looking at the mutations, you're right, the big question always is, are we mapping the epitope or are we just messing up the protein? So how do we ensure that any mutation we look at is maintaining its integrity? So we do that in a couple of ways. One is every mutation we're looking for surface expression, or in most cases looking for surface expression. And we know that if a protein gets to the surface, at least it has to have maintained some semblance of its native-like structure. But we need more detail than that. So what we can often do, what we do as part of the algorithm which we use for doing the epitope mapping, is we compare antibodies to each other. So if we have antibodies that in particular are conformational, that are seeing different parts of the protein, we can use them as internal controls to help us understand whether the protein is still maintaining its native structure. Finally, we can do many types of functional assays. And for example, if a mutation knocks out antibodies but keeps the native function of the protein, then we know that it's maintaining its structural integrity. We've done that a lot for example, viral infection assays. We've also done mapping work on G protein-coupled receptors, where we've done cellular assays for the GPCRs and shown that mutation, which affects an antibody, still maintains function. And of course, this type of analysis can also be used for functional mapping as well as antibody mapping and other applications such as engineering as well.
Great. Thanks very much, Joe. Appreciate that. So our next question is going to be for Jim. Jim, one of our audience members would like to know, do you see any differences in neutralizing binding props between IgG subclasses? I'm assuming most monoclonal antibodies are IgG1.
Well, we certainly do see functional differences, such as different patterns of neutralization or different binding modes when we isolate antibodies of different isotypes. So the isotype that you recover from a human subject when making monoclonals partly depends on the technique. So people using plasmablast sorting or sequencing will often be seeing IgMs because those experiments are done on day 7 after boosting or immunization. Our own group uses mostly memory cells, so typically, as the question asker alluded to, most of those antibodies are going to be IgG1. But certainly we also see IgG2s and IgG3s. Occasionally we'll isolate IgAs from memory cells circulating. And there are several effects you can see. One is the different isotypes have polymorphisms in the framework residues, and those can modulate the binding mode or the angle, or even minor patterns of specificity can change just due to the framework residues. Farther down in the molecule, the isotypes regulate the flexibility of the bivalent binding, or you have 2 FABs in a full-length molecule, so the flexibility can be altered by the isotype or subclass. And then even farther down on the molecule in the FC region of antibodies, some isotypes fix complement better than others, so they'll modulate activity like neutralization. And also the FC region modulates binding to FC receptors, and those are affected by the different glycans that are present in the FC region of the antibody. So those glycans regulate, or they modulate affinity for FcRn, the neonatal FC receptor, which affects half-life. And there are allotypes that are specified by the FC region, and there are recombinant molecules where they're called Ka or LALA variants, where point mutations are made in FC that knock out complement and FCR. So certainly the isotype is important to pay attention to.
Great. Thanks, Jim. Appreciate that. Next question, Lisa, is going to be for you. When discussing freedom to operate, you mentioned that one company might get a narrow patent claim for EGFR antibodies, which is dominated by a broader patent claim by someone else. How can the U.S. Patent Office grant 2 patents that overlap like this?
Thanks, Jeff. That's a great question also. The United States Patent Office is generally concerned with examining applications with respect to the 3 criteria for patentability that I mentioned: utility, novelty, and non-obviousness. So a patent on what we would call a genus, which in this case would be all anti-EGFR antibodies, does not preclude patentability of a species. In this case would be a particular antibody that binds a specific epitope. So the Patent Office will issue 2 patents, and the Patent Office is not concerned that, for example, patentee number 2 with the patent to the specific epitope does not have freedom to operate with respect to patentee 1. So again, the U.S. Patent Office is only concerned with the criteria for patentability and doesn't necessarily concern themselves with freedom to operate.
All right. Thanks, Lisa. Joe, we're going to go back to you. One of our audience members would like to know, do you need the crystal structure for conformational epitope mapping?
Yeah, no, that's a great question. And in fact, the answer, of course, is no. For many membrane proteins, which is a real focus at Integral Molecular, there are no crystal structures for them. And even for many viral proteins, there are no crystal structures for them. And using the shotgun mutagenesis technique, we're able to get information about those residues that are important for the antibody binding. And as I said, if they're not contiguous in sequence, they're almost certainly conformational epitopes. In many cases, for example, for cellular membrane proteins, we can look at snake diagrams, and we can see that the residues we're pulling out are at different parts of the protein in different loops or near different TM domains. What's been interesting is the number of cases we have epitope mapped antibodies seeing residues that were distal to each other on the sequence, and then when the structure has come out, all of a sudden these residues, which are distant from each other in sequence, come together into a compact epitope. And this, of course, is very gratifying and also good validation for the technique. So no, you don't. And in many cases, it's been predictive of what parts of the protein are going to come together.
All right, thanks, Joe. Jim, another question for you. Have you ever seen conflicting epitope map results using different mapping techniques? If so, how would you explain these differences? What would you say is the single most reliable mapping method?
Well, I think the usual outcome is to obtain data sets that don't match perfectly when you're using different techniques. Many of the techniques that I've reviewed are relatively low-resolution techniques. So the hydrogen deuterium exchange only identifies a peptide that is involved in the epitope or even affects the epitope. It's not an amino acid resolution mapping technique. So there's some interpretation based on structure with that type of data, which of the residues in the peptide is likely one that could be involved. Also, some of the EM techniques are, they're not, they're in the 10, 15, 20 angstrom resolution, which again, you can't see side chains there, and you can't see sometimes even specific amino acid interactions. So I think the low resolution nature of some of these techniques leads to some ambiguity. In general, I find that it's helpful if a group trying to do mapping understands some of the biology behind the organism or the target or whatever the antigen is. If you understand the biology, then you can link the functional information you have about your antibodies to the epitope and synthesize all these low-resolution techniques. The other instance where you can see conflicting data is nonspecific binding. So if you just do binding assays, say to overlapping peptides, you may see binding to lots of peptides because there are charge and other interactions that will mediate a nonspecific binding, and those have to be filtered in relation to the other techniques. And also these allosteric effects, when you see a mutation that is not in the epitope but affects the epitope, that can be difficult to interpret unless the data sets from several techniques overlap. And I think it's in the overlap of the data compiled with the biological information. That's where you sort of put it all together.
Okay, great. Thanks, Jim. So it looks like we're going to have time for one last question. Lisa, you mentioned that following several court cases, certain antibody or epitope patent claims may no longer be eligible for patent protection. What happens to all of the issued patents that companies already have obtained claiming antibodies or epitopes?
Thanks, Jeff. That's another really good question. So patents that have been issued are presumed to be valid. So patents with claims to diagnostic tests that under MYRIAD would no longer be valid will not actually be invalidated unless or until a patent holder asserted them against an alleged infringer. And in the same vein, so patents that have claims to epitopes that may no longer be valid under MYRIAD or the ALICE case, they may be ones that a company would not necessarily assert against an infringer and maybe hold back until the laws change if they do. Of course, a patent holder, if they're thinking of asserting a patent against an infringer, should get an opinion from a patent attorney to assess the validity of these claims under the new laws. There's also a question related to bispecific antibodies and their validity. Bispecific antibodies will probably still be patentable because they don't necessarily, they're not found like this in nature, and so if you link a toxin, for example, to an antibody and have the other specificity for a tumor antigen, for example, it may appear obvious, but data could be used to overcome that to show, the binding specificity or avidity or something like that. So bispecific antibodies will probably be a different, animal under these current cases.
Great, thanks, Lisa. And with that, we've come to the end of our webinar. I'd like to remind everyone that the webinar will be archived for 6 months on our website at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Joe, Jim, and Lisa again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful question. And very special thanks to Integral Molecular for sponsoring this webinar. So hopefully we'll see you all again in another Gen webinar in the near future. Goodbye for now.